Hey y'all, congratulations on finishing your quiz. Woo! I'm going to tell you just some housekeeping stuff. First off, hopefully you know that was the last quiz-like thing in biology this semester. What are we doing the rest of the time? It's all in our homepage. So after the quiz, you'll be doing some work consolidating our learning about cells, and it will get us all set up well to dive into some evolutionary topics at the very end of the week and on our last day together of the semester. So we don't have an exam on finals day, but we do have a digital lab to do. And so all this week and on our final day, we have a lot of work to do. Some of it's graded, but no more tests. The next thing I want to do in terms of housekeeping is touch base with y'all about what your notebooks should look like at this point. Before break, you'll remember you had instructions to print out and paste in the pictures you took in microscopy in a specific order. And on our homepage, I colored it somewhere between third period and fifth period, I think, to make it a little bit easier to see, but the information's the same, right? So in your notebook, after the last section on organic macromolecules about carbohydrates, right, where you have your handout and you have your notes, the next thing is your notes on the Edpuzzle with the Miller-Urey experiment and the first protocell. And then after that, you should have a section set aside for bacteria with your picture of the yogurt bacteria, as well as this handout with bacterial cells. If you don't have this handout or any of the other handouts that I talk about, you're going to need them. How do you get them? Well, let's look at today. And it says, pick up or print any one of these printouts that you do not have. If you click on this, it takes you to a page with hot links for all the printouts. So if you're on campus, I may have extras. If you're at home, I can't get it to you so easily. Almost all of these you should already have. If you're in fifth period or seventh period, there are a couple here that you're new ones that you're going to need through the course of the work you're about to do. All right, so again, your notebook should have a section, the cell section after the Edpuzzle notes should have a section about bacteria. Next, you should have set aside a section for archaea, which we will learn about. Next, it should have your protist notes. It should have the dichotomous key for identification. It should have your pictures. It should have, ideally, the graphics that were given to you so that you can look at your picture as well as a simplified version of how people envision it, as well as any other notes about protists. Next, you should have a section for fungus with your picture of yeast. First and third period already have this picture of fungal structures. Fifth and seventh period, you'll either need to print it yourself or pick it up from me if we're in person. And you're going to end up taking a fair amount of notes, all of y'all, no matter what class period, that you're going to end up adding quite a few notes to your fungus section, which is great. That's why I told you to save a 2-page spread for each section. After fungus, you should have plant notes with your pictures from microscopy and this graphic of a plant and animal cell. Your pictures should have identifications as best as you could of the different structures. And then you should have saved a section for animal cells. Now then, if you're in first or third periods, verify that your notebook is in good order. You will already have this scale document and this tree of life pasted in with notes. So if you're in first or third period, I'm about to talk about things you've already heard about, specifically the Miller-Urey experiment, the first protocell. We're going to touch base on the scale of relative sizes of things and paste that in our notebooks and talk about the tree of life. So you've already heard all that. You can go to the next thing in the module, which is an assessment. You'll notice that you can't move on until you get 100% on this. Everything is in your notes, so you'll do great. If you don't get 100% the first time through, you maybe should watch the rest of the video, fill in your notes, and then do it again. If the second time through you or anybody else doesn't get 100%, then you need to come talk to me and we'll see what's going on. After you finish this series of questions, once you've got 100 on that, you can move on to the next part of the module. The next thing will be plants. Again, first and third period, you can skip that video because we've already talked about that, and you can go directly to the assessment. All right, and if you have questions, just let me know. All right, so if you're still here, it means you're excited to review and look at what we learned about going from macromolecules to cells. So the notes you took about that were from the Edpuzzle with Professor Dave. Woo. So the first thing it says focused on was the Miller-Urey experiment, and it's talking about this experiment that was conducted in the 1950s. It was trying to mimic Earth, and during what time period? Well, around 4.2 billion years ago. Not bouillon. 4.2 billion years ago. Some people ended up putting 4.4. They start talking about 4.4, but he says at 4.4 billion years ago, the Earth was way too hot. By 4.2 billion years ago, however, things had cooled down enough for what? Why did it need to cool? It needed to cool enough for liquid water to form. And in fact, this is when oceans formed. Then you needed to fill in your schemata of the experiment. So it should look, yours should look something like this, right? Down here was a little flask with water, because again, liquid water. And there was heat because, again, Earth was hot, and also the sun. And that evaporated water. The water vapor went up the tube and around and then into this flask, which simulated the Earth's atmosphere. And the Earth's atmosphere, to the best of their knowledge, had methane, ammonia, hydrogen in it. Since then, there are different ideas about exactly what gases there would have been, but the premise still stands. And this experiment's been repeated several times with many different variables with very similar outcomes. In that mix, they added an electric current to simulate lightning. So in that atmosphere, it was a very volatile place. We didn't have to label it, but right here was a condenser. That means there was sort of an area with cold water running around it, not in it. They didn't add water at this point, but just around the tube, like cold water just circulating to cool it down. And when vapor cools down, it condenses. So that's why you'd have liquid dripping down. And here is where they collected to take samples to see what formed. And within 1 week, amino acids formed, right? Whereas on Earth, you had a time scale more like 500 million years for this volatile situation to happen. So if you had amino acids within a week, one can infer with good evidence that those volatile molecules would bond to one another. Amino acids would bond to each other, forming proteins. Enzymes, which are kinds of proteins, are enzymes to catalyze reactions, which would speed up and start even more chemical reactions. Those enzymes might help form DNA, RNA. So this experiment shows that the molecules of life, the molecules that build living things, were formable on our planet during the time at which it seems life started to evolve. So it didn't take a magical event, pixie dust or something, for life to emerge. We have evidence that the building blocks of life were formable on our planet at that time. The next thing in your notes was to sketch out just that first protocell. So what would that kind of bridge structure be going from the molecules that could form to a cell? What did it look like? Some folks seemed very concerned about labeling every little bit of the picture that the video had. I just wanted, which wasn't the intent, I just wanted you to have a sense of what would be enclosing this protocell and what might be inside. Well, we just talked about what might be inside. Amino acids, proteins, DNA, RNA. We have evidence that those kinds of things could have formed. And then around the outside, it would be a phospholipid bilayer. All right, so there are two other things we're going to do. in this video. One is we're going to add to your notes the document that has a scale of sizes. So if you're in the classroom and you can pick one up, go ahead. It looked like that. If not, in the link, which is from our homepage, that has all the handouts, it's the one that says scale showing relative sizes of things. All right, cut that out and paste it into your notebook after the animal cell section. So you should have set aside some space for animal cells that we'll use later. The next page, you should paste that document and then just take some time to look at it, build familiarity with the units used, with what's visible with an electron microscope as opposed to a light microscope. We, of course, were using light microscopes. So from that and looking at it, sometimes we were wondering, well, what am I seeing? Could I see a protein? Could I see a virus? No, those are way too small for a light microscope. But there are plenty of things that we could and did see. Once you've got that pasted in, take a look at it. Notice that it tells you units on the left-hand side of the scale. It tells you what units they're talking about, just to get oriented. Then the next thing we're going to talk about, and some classes did already, so you may have already got this part. You may just need to paste it in. But the next page should have the Tree of Life and notes about the Tree of Life. So if you need that Tree of Life, again, print it from this page or pick it up from me. It's well accepted in the scientific community that life be chunked up into 3 groups, and those big groups are called domains. So in your notes, you should have that they're generally agreed upon 3 domains: bacteria, archaea, and eukarya, or eukaryota. You notice that bacteria and archaea are prokaryotic and eukarya are eukaryotes. You'll also hopefully notice that animals, fungi, and plants, the things we think about mostly as living, are just this tiny little part of the tree. We are by no means the majority of life on this planet, not even the majority of eukaryotes. And there's a lot of other stuff that's eukaryotic. You'll recall from our pathogens unit that all the other eukaryotes that don't fit as animals, plants, or fungi are lumped together as protists. So go ahead and circle on your Tree of Life the protists and label them protists. Process the fact that this is really a hot mess of a group, right? This would be worse than even just saying to you and a portobello mushroom and an oak tree that you're all basically the same, right? Because a slime mold up here is genetically and structurally less like diplomonads than you are to a mushroom. You are more similar to a mushroom than they're similar to each other, and yet we dump them together. So it is a problem. That said, it's convenient. So we have an additional layer of organization under domains that it's convenient. It's certainly not fully agreed upon, but we'll use it, of 6 kingdoms. So those kingdoms: bacteria, which corresponds to the domain; archaea, which corresponds to the domain; and then fungi, plant, and animal, which we're kind of familiar with; and protists. All right, so if you have this in your notes, that is the last bit for this video. Woot! Go and answer the questions in the module and just move along the module and take little breaks between each one, because don't sit for the whole time.